Thank you very much for this kind introduction and for the opportunity to present my work in this nice Sussex AI seminar here. The topic of today is a neuroscientific benchmark model, optimizing for accuracy and efficiency. But first, a few words about myself. So I studied physics in Germany at RWTH Aachen University and in Italy at the University of Trieste. And then I moved for the large part of my research to Jülich Research Center, as Thomas already introduced. And nowadays I'm there, the team leader of the team Future Simulation Architectures, and joined recently the University of Sussex as a lecturer in computer science. My research interests are concerned with network models of cerebral cortex and simulation technology. I will, in the end of my talk, come back to the models I'm currently working with. Yeah, so as you all know, benchmarks drive AI research. This here is a figure from Stanford University that shows the performance of AI systems relative to human performance from in the last decade. And it shows that human level performance has been reached on certain benchmarks. For example, for image classification or language understanding. But I have to say, just for certain well-defined benchmarks and at very high energy cost and with models and learning mechanisms that are most likely very different from biological neural networks. I'm from the field of computational neuroscience, and we work with constructing, simulating, and analyzing models of biological neural networks. Why do we need benchmarks in this field? And with benchmarks, I would lose this term a little bit, use a broader scope, and just say, with it, we need some systematic comparison. For example, we need it if there are different implementations of a model and we want to ask, do we simulate the correct mathematical model or a correct model of the biological counterpart that we want to learn more about? Or if there are different software and hardware environments, like high-performance computing systems of the conventional type, or also more dedicated neuromorphic hardware, do we make best use of our resources? Do we actually simulate as efficient as we can? And so these are reasons why benchmarks are interesting in this field. What do we mean actually with simulations in computational neuroscience? And this is a little example indicating how our simulations somewhat mimic neuroscientific experiments. So here are two neurons, a presynaptic and a postsynaptic cell depicted. They are connected by a synapse. And if you, for example, stimulate the presynaptic cell by introducing some current to it, we simulate its membrane potential, and that rises in response to this input until it eventually hits a certain spiking threshold, and that's when the neuron emits a spike, or then, if this happens several times, a spike train. These are just stereotypic events in time, and that's how the neuron communicates its information to a postsynaptic neuron. Recording there would result in measuring a postsynaptic response in that neuron's membrane potential. So this is in principle what we simulate at the very basic of these neuroscientific simulations. And a simulator that we're using here as a reference is called NEST. So there are many different simulators, software-based, hardware-based, that function in a similar way. And so here we have depicted this one that aims at simulating spiking neural networks with a focus on the dynamics, size, and structure of neural systems rather than on the exact morphology and details of individual neurons. The simulator uses a C++ core, parallelization with OpenMP and MPI, but for defining the simulations, there's a Python front end called PyNest. And in principle, the same code can be used from laptops to supercomputers. So you can define your small networks and run them on your laptop, or you can go to really large networks and that require supercomputing. The development of NEST is driven by scientific needs with a focus on accuracy, flexibility, and quality assurance. And to the NEST ecosystem, there belongs also since recently NEST GPU, a version that draws its power from not only from CPUs, but then also from GPUs. There's NEST Desktop, a graphical user interface for this. And there's NestML that allows you to define your own models. So there are many pre-built models, but you can also define your own ones with that. Okay, so this is the simulation environment that we use for reference. And now we come to the models. So this here is a model of a cortical microcircuit. It captures about 1 square millimeter of cortical surface area. It represents 4 cortical layers, 2, 3, 4, 5, and 6. And in each of these layers, there's an excitatory and an inhibitory neuron population. That means excitatory neurons drive the postsynaptic potential closer towards the spiking threshold, and inhibitory neurons farther away from that. Yeah, so this model has in total 80,000 leaky integrated fire neurons that have a similar spiking mechanism as the one that I just showed to you, and about 300 million synapses or connections. So it's already this is the realistic neuron and synapse density for this 1 square millimeter roughly. Yeah, and the connectivity of the model is derived from experimental data, so it takes into account layer and neuron type specific connectivity, and this is sufficient to reproduce experimentally observed dynamics. And I just showed to you what a spike train is. So this so-called raster plot shows in each row a spike train over time of one neuron, and here you see for the 4 different layers, the spikes of excitatory and in black and inhibitory neurons in gray. Yeah, so and this model already reproduces asynchronous irregular spiking of neurons. So we also call that AI, meaning in that sense asynchronous irregular. The spike rates of inhibitory neurons are usually a bit higher than of excitatory neurons. And the distribution of spike rates is also somewhat correct across the layers. So it's, in that sense, a quite minimal model, but it already reproduces some of the experimentally observed spiking activities. Yeah, and this model has actually become some kind of a standard benchmark model in the past years. And we think that 2 reasons are because of its relevance and of its feasibility to use it as such a model. So we thought that a neuroscientific benchmark model should account for brain structures and dynamics at realistic spatial and temporal scales. The model should represent some fundamental, generic, and sufficiently understood biological mechanisms. And it should provide potential for extension, for example, for upscaling or for inclusion of more complex features. In addition, this model has only moderate hardware and software requirements, and that means it enables routine simulations with off-the-shelf computing systems. Yeah, so going back a few years, we asked quite naive questions. That was if a simulation of the same neural network model is run both on an HPC, high performance computing system, using the simulator Nest, and on the neuromorphic hardware system Spinnaker, are the results the same? So we set up some kind of workflow for comparing Nest simulation running on a rather conventional high-performance computing system with a simulation of what we thought the same model on a Spinnaker, which is the neuromorphic hardware also developed here close by in Manchester, that is based on ARM chips. And we try to set up some simulation analysis pipeline to compare the results. So really quickly we realized that the answer to this question was no. So first of all, we had not defined what we actually mean with simulation, or what we mean with the same neural network model, or what is actually the HPC system in terms of its hardware, software, environments, and so on. And also the simulators have different versions or different compatibilities and so on. And for the analysis, what do we mean with results? How do we want to analyze this main outcome that are just these spike trains? And are we happy when can we say results are the same? So there was more depth into that. And in the following, I would like to show you some points on the accuracy of this model, how we try to compare it, how we try to understand and adapt the model in the afterwards, and how we went on to measuring also the performance, the time, and the energy, and to our steps towards more systematic benchmarking and models beyond the small microcircuit. Starting with a comparison of single neuron dynamics. What you can see here is the membrane potential of a neuron simulated with Nest, grid-based, so with a different time step, 0.1, what we mostly use milliseconds, or 1 milliseconds, and the neuron receives stimulus with a rather low rate or a high rate, so low rate in the at the top and low rate at the bottom. And the different curves that I show here are ones from this grid-based Nest, and in blue with with Spinnaker in red, and the dashed black line is the precise Nest that we use as reference, so in which the spike times are not restricted to a grid. And what you can see here already is that Spinnaker always comes a bit is a bit slow, and the grid-based Nest is a bit too fast when you compare with these spike times. And so with this, I want to show that also a priori we do not expect exactly the same results. So, for example, there are certain differences between the simulators. Nest uses floating point points, and Spinnaker fixed point numerics. Nest uses an exact subthreshold integration of the dynamics, and Spinnaker uses a separate integration of membrane voltages and synaptic inputs. And what matters then for when we go from the single neuron to a network is that there are also different random number generators. So let's go to the network dynamics. Here I'm showing raster plots of left the grid-based Nest, in the middle the precise Nest, and on the right Spinnaker. And as you can see is that it's very, very hard to interpret this by eye. So we have to go to more sophisticated analysis method. We can compare these only on a statistical level. And what we usually do is that we calculate for each of these 8 populations, for excitatory and for inhibitory populations, measures like the spike rate, so a spike rate per neuron, number of spikes divided by the time interval with, and also coefficient of variation of the interspike intervals and correlation coefficients. So we calculate all these measures and look at the distributions, and here you can probably only see rather one line for each, and that means that at that point we had achieved the point that all these curves lie one on top of the other, and we were somewhat happy with the results being the same in that sense. Yeah, but it took a while until we got there, actually. Yeah, so about understanding and adapting the model further, we scrutinized some model assumptions. We wanted to see if what we did in this very first attempt, if this was all valid, and check. For example, here we simulated in this first study only 10 seconds of biological time, and if we take the spike times from the simulation and randomize them, so we generate some kind of surrogate data and try to calculate the same measures here with this surrogate data, something surprising came out, and that is for the correlation coefficients, the actual data, the reference data and the surrogate data, they were just indistinguishable. So that means that there in this measure of the correlation coefficient, there was no information about the actual network dynamics if we analyzed just such a little bit of data. So this was more like a finite size effect that we measured here. So let me put this right. If something really went wrong, we would have detected it. If there was some difference, but if the exact spike times were taken into account, we would not have seen any difference. So we looked at how long, what is actually the role of observation duration, and if we simulated for like 15 minutes, we noticed that there was then a clear difference between the reference data and the surrogate data, and from then on we only simulated much longer to get also some meaningful result for this measure. Yeah, and another thing that how we tried to investigate the model further was we looked at connectivity concepts. So here is just an example from some toy network where a presynaptic neuron population in green is connected to a postsynaptic population in orange, and with connectivity concepts we mean how is this connectivity between the pre and the postsynaptic population defined. So this could be one-to-one or all-to-all, like some deterministic rule, or it could also be a probabilistic rule, like a fixed in degree, meaning that each neuron from a postsynaptic population receives the same number of inputs from the presynaptic population, but here the sources are randomly chosen. In the cortical microcircuit model, actually a random fixed total number is used. That means the total number of connections is kept fixed, but how it is distributed on pre and postsynaptic neuron is a random process. In the study I'm citing here, we actually investigated how the descriptions of connectivity in different articles, how well connectivity was described in different articles of computational models, and we found out that a quite large proportion had some ambiguity in the description of connectivity, and in that study we then provided some guidelines for connectivity concepts, meaning for specifying connectivity in a complete and concise way, together with a graphical notation for network diagrams that we proposed. Okay, so but coming back to this microcircuit model that uses per default the fixed total number connectivity, there we noticed that if the indegrees were sufficiently heterogeneous, as it was in this default case, the firing statistics could be preserved even if all synaptic weights were replaced by the mean of the weight distribution. So it uses a normal distribution of weights per default. So with weights, I just mean the connection strength here. And there we noticed if we just fixed them, that would also work well enough to reproduce the statistics of this model. And this could be a nice insight if one wanted to bring the model to hardware, for example, where not every weight can have a different value. So, this was not the case if the indegrees were kind of homogeneous. So we tested this with the fixed indegree version of the model. And another thing that we tried to understand the model better was to use some mean field theory. So we have published a mean field toolbox about that is a Python module with which one can make predictions about the activity, about the dynamics of the model, and directly compare them to the simulation. So, for example, for the firing rates of the neurons in the different populations, we could then directly compare the bars that are from the simulation with the theory calculated with that toolbox. And another thing one could also investigate with such measures is if there are some oscillation in the activity by calculating, for example, power spectra and finding out at which frequencies are peaks visible. And there is a measure for identifying then the anatomical origins of coexisting oscillations. So there's then a method for finding out which of these connections between these neuron populations are responsible for the amplitude and the frequency of certain oscillations. Here I would like to mention that the activity, that the connectivity in the model, as I said, comes from different experimental sources, but there's also room where we do not know if the estimates are correct because that experimental data was incomplete, and in that sense one could use such mean field methods as some kind of dynamical argument to understand better if the connections chosen for the model make sense or potentially need improvement. Yeah, so in that sense, we have some more understanding about the model. Now I've talked a lot about the accuracy, about some model details, but as for the benchmark, the other aspect is also important. That is, what is actually the time to solution? How fast can we simulate that model? And how much energy does that take? Yeah, so I'm coming back now to this Nest-Spinnaker comparison, this first study I started off with, and what we did there, we actually measured directly at the compute nodes how much power does the simulation take, and we could measure here, for example, the power versus time. The red part here for the Nest simulation was the part when the network was constructed in computer memory, and the blue part here is when the simulation was actually running. And so this here was on one. So this middle panel was on one CPU, and then on two CPUs. The the simulation is shorter, but the power is also higher. And at that time on Spinnaker, the whole part of of connectivity data generation and loading it into the model took a couple of hours. In subsequent improvements, this was then not the case anymore. So we could also then go from this to a so-called strong scaling experiment, where we not only looked at just one CPU and simulated the model just on one core and subsequently to all cores of one CPU, and then adding more and more hardware to it in a sense. And what you can see here is in blue for the nest simulation, the simulation time that decreases until it's saturated at some point. In red, there is the network construction time that also decreases when parallelized across more hardware. And the black line here gives real time. So with Nest at that time, we achieved about 2.5 times real time. That was the fastest we could get. And for Spinnaker, they had to slow down. And the main reason for that was this time step of 0.1 milliseconds. So we needed that resolution to resolve the fast neuron dynamics. And they had to slow down by a factor of 20, but I can already say that soon later they actually achieved real time. And while we were measuring these times, we were also measuring the power and with Nest, and this horizontal line is with Spinnaker. So the actual energy that simulation took in the end was then for the fastest cases quite similar again between Nest and Spinnaker. So Nest reaches real time a couple of years later, and even sub-real time, and we usually measure that with the so-called real-time factor. That's how we define a factor being the wall clock time measured divided by the biological model time. And this has, so the reason why Nest has become faster is because on the one hand the software was further optimized, but on the other hand it was also the hardware that got replaced at some point. So one has to think about these two things always, of course. But otherwise, more or less a similar experiment was done in this subsequent study. So after this first benchmarking experiment, I would like to show you how we started to do this in a more systematic way. And there was a study in which we first on a rather conceptual level thought about which kind of dimensions does such a benchmarking experiment have? What do we have to track, to take care of, to also which metadata should we capture to make this reproducible and understandable? So that the dimensions that we have here are the hardware configuration, software configuration, the simulator details, and then the details about the model and the parameters. And last but not least, also who did these benchmarks. So communication between researchers also played a large role if we wanted to understand what someone else has done at some time before. And we set up a benchmarking framework called Bench, and with which we can measure then we do strong scaling, or also weak scaling experiments, in which we measured the network construction time or the state propagation time. At that time only with NEST, and could see how these improve when a simulator was developed further. So, for example, here this was an older version of the simulator NEST when this microcircuit model was simulated. So the vertical axis is the time, and the horizontal axis is the number of compute nodes, so in principle the hardware used, and the blue is the network construction time, and pink the state propagation time. And as you can see, the state propagation time went up at some point, and we figured out that the reason was communication, so MPI communication between the compute nodes played a role that could be fixed and improved, so that in the new version this curve was getting much flatter. So in that sense, we were trying to implement more and more systematic benchmarking in the development of the simulator, for being able to track our benchmarking details, and also for being able to compare this better to other simulation environments. And it was actually not just NEST that worked on that, but also other simulators. And here's some kind of summarizing slide, which I want to show you this real-time factor. This gray line in the middle of the plot is real-time. And the horizontal axis is the years from 2018, when we had our very first study, to last year in 2023. And the different data points here show the different simulation technologies. So you can see, for example, Spinnaker here in the beginning in our very first study, with a real-time factor of about 20, but soon after already reaching real-time in another published study. You can see the CPU version of NEST also making some improvement from the two studies that I showed to you. There is also the GPU version of NEST showing some results, and another GPU simulator called GEN that is also developed here, that outperformed NEST GPU here as well. And but nowadays the fastest simulation that were published about this cortical microcircuit model, they were run on an FPGA system actually. So on an actual FPGA cluster. So with this, I want to say that this was actually some kind of performance gain then for the whole community, for different, for the developers of different simulation technologies, who all tried to implement that model to their system, trying to find out where the limitations are. So, for example, in terms of the requirements, like the number of synapses per neurons, or this simulation time step that are required for resolving these fast dynamics, and a lot of very interesting and useful algorithmic solutions have come out of this. So it's a performance gain by 2 orders of magnitude in the simulation speed, and also for the energy consumption. I can only say at this point that there have also been some improvements. So the model of the microcircuit is just some kind of a starting point. It is a very small and somewhat simple model, but it has the potential for extension. And I would like to show you two models that build on that. And one is here a multi-area model with 32 vision-related areas of macaque-monkey cortex. And this model was in a similar way as this microcircuit model developed and first tested with a nest simulation, and but soon after with GEN and also nest GPU results were obtained and compared. So maybe a few more words about this model. It actually uses as each of these 32 areas one version of this microcircuit model adapted to the different areas in terms of their different connection probabilities or neuron densities and so on. And so that one can have the activity in different of these different areas showing different characteristics. And the model is also tested in different network states, like a ground state and a more metastable state, in which the network switches is not purely asynchronous irregular, but switches between phases which the network is more active or less. And there are also variations of this model in development. So recently accepted a version of how it was adapted to the human cortex, actually. And another study I was involved in, I wanted to show briefly to you, is this model from my second slide. So this model here is a model that laterally extends this microcircuit, and it extends it to a size of 4 by 4 square millimeter, which is exactly the size of a Utah array, that is a multi-electrode array with 10 by 10 electrodes that are used for electrophysiological recordings. And the idea of the model was that we get a model that represents exactly the cortex at realistic neuron and synapse density at the scale, allowing then for a direct comparison between model activity and recorded data. And what is new in this model is not only that size, so it has then in total 3.2 million neurons and 7.7 billion synapses, but also that the neurons are actually arranged in space, which was not the case with the original microcircuit. So the neurons have 2D positions, and the connectivity between them is distance dependent. So we take into account that neurons that are closer to each other have a higher probability of being connected than neurons that are rather far away from each other. And there are then also like distance dependent delays and so on. Yeah, and we are analyzing in this model the spiking activity, in addition, we predict via a forward modeling approach local field potentials. So the recordings of this Utah array, these are extracellular recordings. So these electrodes are in the space between the neurons, and the low-pass filtered signal that one can directly measure from it is called the local field potential, so a signal to which several neurons contribute, and the high-pass filtered signal would be the spiking activity. So with this model, we can then also mimic measurements at 10 by 10 electrodes in the same way, and we use this forward modeling approach by mapping the spike trains of the point neuron simulation to morphologically detailed neurons to predict the current going in and out for the local field potential. And the result of this study is that we have seen, similar to experiments, that correlations in spike trains are very weak, so close to zero, and there's no distance dependency. The y-axis in this figure here is the distance, but if we have then these signals to which multiple neurons contribute, we can see that in the correlations of the local field potentials have then this distance dependence in the correlations that is not visible in the spike train correlations. And this is also comparable to experimental data. So this study has also just been accepted and will be hopefully out very soon. We have also made the whole model code public and available for trying out. So now I come to my summary. So what are the lessons that we have learned in this whole process, and what kind of outlook can we give? So the benchmarks require cross-disciplinary co-development of neuroscientific models and simulation technologies. And with this, I mean that there are the neuroscientists who try to understand the biology, to think about which parts of the biological neural network should be modeled in which way, which requirements are there in terms of, let's say, timescales or connectivity, and this should be then in close contact together with people who work on simulation technologies for neuroscientific research, or ultimately for would also be interesting for neuromorphic hardware trying to become more brain-like, making use of advantages like the sparse encoding in the brain. But for the benchmarks, we also need unambiguous model definitions and benchmark settings, so clear definitions of how a model is mathematically defined, independent from the algorithmic version that is then implemented into some simulator. The model needs to be clearly defined, but also the benchmarks. For example, which times do we actually compare? Usually there's some kind of startup transient in such a simulation that should be discarded for comparing, for example, the activity of the models and so on. It's also important that there's an understanding of the model characteristics and the performance metrics. So dependent on the model that is used, there are probably different metrics that are of interest that should be preserved, even if there are different simulations, and need to be defined on a case-by-case basis. Then it's important to define some kind of reference technology and useful means to compare technologies. That there are, for example, also independent hardware and software improvements, as I showed to you, that there were these improvements of just a simulator nest a few years later, and then there can be some creative algorithmic solutions. And then with this talk, I wanted to show how important it is to build on each other's work. So on reaching and surpassing real-time simulation was here possible in only a few years by the series of publications around this one small model. And last but not least, that it's also important to go beyond that, to so these simulation technologies are usually intended to be quite general, so they should not be fine-tuned to a certain model, and therefore it's also important to have rather a battery of benchmark models when using them to optimize the technologies. And in addition to having different models, it's also important to go to more complex models, so to upscale from the local to brain size networks, or also to take into account functional models that come with their own performance metrics, for instance. So, and with this, I would like to end. I would like to thank you very much for listening, and I would like to thank my colleagues here at Sussex and at Jülich Research Center and the external collaborators that contributed to the studies that I have presented. So, thank you very much. Are there any questions? Yes, please.
You had a nice slide showing the results of the benchmark, basically, and how the different systems were comparing to real-time, real-time vector. Did you also manage to gather data on the other thing you mentioned at the beginning, the power consumption?
Yes, I didn't want to show this such a figure yet because the data that I have there is not directly comparable, because we sometimes it depends on how the energy is measured, right? So we usually measure energy, or we calculate energy per synaptic event, and with this we mean in principle like spikes going through the network. But sometimes people measure, as we did, directly at the compute node. If maybe some have included things like an interconnect. And so I did not include this because it was not directly comparable yet. But there is of course also an improvement, not only by the just reduced time that was used, as energy is just the integrated power over time, and I don't have the data to show you.
Thanks, Johanna. That's nice talk. So you're focusing on performance metrics and the concept of a good model, and I guess a model is good in the context of a particular scientific question. I'm interested in what kind of scientific questions do you think you can help get answered with these kind of large-scale models, given we're still a million miles from theoretical models of the brain?
So I think that we need this co-development of the neuroscientists asking the questions, and the simulator developments trying to make possible answering the questions. And so, for example, there are many phenomena like plasticity phenomena that are at the moment very hard to study in large-scale networks, just because the simulations for these really large networks cannot be done in real time. They're usually much slower. So I think there are usually models for different scientific questions, and one has to see how with which simulation technology they can be simulated.
So phenomena like plasticity, do you think for me it feels like a small network, it's easier to figure out plasticity rules in a sense.
Yes.
But are there advantages to large networks with that, or any other kind of stuff with it?
So it really depends on the scientific question, I would say. So for example, if you're simulating large networks as they are in the brain, it's not possible to downscale them while preserving at the same time all the correlation structure. But so in that sense, it's important if you want to measure all the interactions between different brain areas, for example, to have that full scale. But if one's interested in studying a mechanism with some which let's say fewer neurons contribute, then this such a functional network. I think then it of course makes total sense to have this as some kind of complementary model to test as well.
Thanks.
That makes sense.
Yeah, thanks for the talk. So following up from that question, has anybody actually implemented plasticity rules in this model? And if they have, are you able to provide an example of the kind of task that they gave the model, or what it was that they were trying to get the model to learn?
So I think that there was, I think with this particular model, there was, I think, no study published that was really focusing on a certain size. So there are actually quite some studies looking at some visual phenomena also, but and I remember a study where they focused on where they put STDP in, but I think it's all not it's not well studied yet with respect to STDP or other plasticity mechanisms.
And there is that Wolfgang Master paper where they train different microcircuit models end to end with backup for time on tasks that mice are trained on, which is kind of interesting.
Yeah, that's not.
But it's not this model.
It's not this model, no. Yeah.
I have a big question. I don't know whether you can go back all the way to the slide with the small scaling, but this one, that was great. So, what's happening there, just out of curiosity, with this break in your axis?
These.
These lines.
Okay. So that was a machine where we had CPUs that had 24 cores, and then from 24 to 48, this was hyperthreading, so using two threads per core. And then we were adding CPU, whole CPU. I think always groups like four additional CPUs. Yeah, so this was actually done on a cluster with 32 compute nodes. We had full control over the system and could really switch on one after the other.
And you divide the red line and top is jumping at 16. Network construction. That seems.
Yeah, I think we have not. Yeah, I think we have not fully understood this at that time. Yeah.
It's funny, yeah. Thank you.
Yes.
So in performance metrics, you're thinking about actually statistics, right? Like correlations between neurons and stuff, right?
Yeah, but the reason for that was just that this model was mainly focusing on dynamics, not a function, and so then we thought that statistics.
I guess I was just asking, would there be an interesting way to, or an interesting function, that how can you add a particular function, or what kind of functions might be relevant for this kind of model?
Yeah, so related to the question, so I think what one could potentially do is to provide some input to the model and see how it reacts, also to some structured input, do some functional task with it. So this model has built in like a very simplified thalamic input going to layer 6 and 4. So this could be the pathway for feeding in some signal to analyze then.
So the function being that you put an input signal and you see that the statistics of the output are similar to the answer.
That's possible. Any other questions?